Hi, Gina Legrand here. Thank you for taking the time to view my presentation on oxygen binding in heme. I hope you enjoy it. What is hemoglobin? Hemoglobin is a protein molecule found in red blood cells made of 4 subunits, 2 alpha subunits and 2 beta subunits. Each subunit surrounds a central heme group that contains iron and binds 1 oxygen molecule, allowing each hemoglobin molecule to bind 4 oxygen molecules. Oxygenated arterial blood, where hemoglobin is transporting 4 oxygen molecules, is bright red, while venous blood that is deoxygenated is darker red. How does oxygen bind to heme? The process of oxygen binding by hemoglobin is comparable to substrate binding by an allosteric enzyme. In the laboratory, hemoglobin can be extracted from red blood cells and fully purified. In this purified state, its affinity for oxygen is much greater than what is found in red blood cells. In fact, the affinity is so high, only 8% of the oxygen would be released to the tissues if hemoglobin in the cell operated the way that purified hemoglobin operates. Under purified conditions, hemoglobin would be incompatible with supporting life because it would be unable to release oxygen. So what makes the cellular environment different? Within red blood cells lies an allosteric regulator molecule called 2,3-biphosphoglycerate, which we'll refer to as 2,3-BPG. This highly anionic compound is present at approximately the same concentration as hemoglobin. When bound to hemoglobin, it reduces oxygen affinity so that 66% of the oxygen is released instead of a scant 8%. How does 2,3-BPG affect oxygen affinity? When we examine the crystal structure of deoxyhemoglobin in the presence of 2,3-BPG, we see that a single molecule of 2,3-BPG binds in a pocket at the center of the hemoglobin tetramer, which is only present in its T form. This interaction is facilitated by ionic bonds between the negative charges on 2,3-BPG and 3 positively charged groups of each beta chain. 2,3-BPG binds to deoxyhemoglobin and stabilizes it, reducing its oxygen affinity and causing it to release oxygen. For the structural transition from T to R to take place, the bonds between hemoglobin and 2,3-BPG must be broken, and 2,3-BPG must be expelled. Structural changes in hemoglobin. Like all allosteric proteins, hemoglobin displays a quaternary structure. As stated earlier, human hemoglobin A present in adults consists of 4 subunits, 2 alpha units and 2 beta units. The alpha and beta subunits have 3-dimensional structures that are similar to myoglobin and are best described as a pair of identical alpha-beta dimers that associate to form the hemoglobin tetramer. From tense to relaxed. How does oxygen binding lead to the structural transition from the T state to the R state? When the iron moves into the plane of porphyrin, the histidine residue bound in the 5th coordination site moves with it. This histidine residue is part of an alpha helix, which also moves, which we could see in the first image. The carboxyl terminal end of this alpha helix lies in the interface between the 2 alpha-beta dimers. Consequently, the structural transition at the iron ion is directly transmitted to the other subunits, resulting in substantial changes in quaternary structure that correspond to the T to R state transition, which can be seen in the second image. The A1B1 and A2B2 dimers rotate approximately 15 degrees with respect to one another. The rearrangement of the dimer interface provides a pathway for communication between subunits. The presence of oxygen on one of the subunits is immediately communicated to the others so that the subunits change from T to R in concert. The binding capacity of oxygen to heme in the lungs. The primary activity in the lungs is the process of respiration, or gas exchange. The function of respiration is to provide oxygen for use by body cells during cellular respiration and to eliminate carbon dioxide, a waste product of cellular respiration from the body. For the exchange of oxygen and carbon dioxide to occur, both gases must be transported between the external and internal respiration sites. Although carbon dioxide is more soluble than oxygen in blood, both gases require a specialized transport system for the majority of the gas molecules to be moved between the lungs and other tissues. In the lungs, hemoglobin becomes nearly saturated with oxygen, such that 98% of the oxygen-binding sites are occupied. When hemoglobin moves to the tissues, oxygen saturation level drops to 32%, so 66% of the potential oxygen-binding sites release oxygen to the tissues. Capillary gas exchange. As blood passes through the lungs, the hemoglobin picks up oxygen because of the increased oxygen pressure in the capillaries of the lungs, and can then release this oxygen to body cells where the oxygen pressure in the tissues is lower. In addition, the red blood cells can pick up the waste product, carbon dioxide, some of which is carried by the hemoglobin at a different site from where it carries the oxygen, while the rest is dissolved in plasma. Even though oxygen is transported via the blood, you may recall that oxygen is not very soluble in liquids. Only 1.5% of total oxygen can be dissolved in the bloodstream. Most oxygen molecules are carried from the lungs to the body's tissues by a specialized transport system, which relies on the hemoglobin present in blood cells. As oxygen diffuses across the respiratory membrane from the alveolus to the capillary, it also diffuses into the red blood cell and is bound by hemoglobin. The reversible chemical reaction, hemoglobin plus O2 yields hemoglobin minus O2, describes the production of the final product, oxyhemoglobin, which is formed when oxygen binds to hemoglobin. Oxyhemoglobin is a bright red-colored molecule that contributes to the bright red color of oxygenated blood. In this formula, Hb represents reduced hemoglobin, that is, hemoglobin that does not have oxygen bound to it, as you can see in this equation. CO2, water, and pH. The presence of carbon dioxide gives rise to the release of oxygen from hemoglobin. The first way it does this is that at high concentrations, carbon dioxide reduces pH. This occurs when CO2 reacts with water and forms carbonic acid, which dissociates to release H+ and bicarbonate ion, ultimately decreasing pH. This reaction is sped up very quickly with an enzyme present in red blood cells called carbonic anhydrase. The second way CO2 impacts the release of oxygen is the direct interaction that CO2 has with the hemoglobin itself. CO2 stabilizes the deoxyhemoglobin form by reacting with terminal amino groups to form a carbonate group, which is negatively charged. These negatively charged groups participate in salt bridges. The presence of salt bridges stabilize the T state, driving oxygen to be released from hemoglobin. The Bohr effect. To tie up our discussion about the effect of CO2 on oxygen binding, I wanted to briefly touch on the Bohr effect. The Bohr effect is a phenomenon that arises from the relationship between pH and oxygen's affinity for hemoglobin. A lower, more acidic pH promotes oxygen dissociation from hemoglobin. In contrast, a higher, more basic pH inhibits oxygen dissociation from hemoglobin. The greater the levels of CO2 in the blood, the more molecules that must be converted, which in turn generates hydrogen ions and lowers blood pH. Furthermore, blood pH may become more acidic when certain byproducts of cell metabolism such as lactic acid, carbonic acid, and carbon dioxide are released into the bloodstream. For my final slide and to wrap up the presentation, I thought it might be interesting to share a little about my own experiences with an inherited blood disorder. When I was 35, I was diagnosed with beta thalassemia, an inherited blood disorder characterized by reduced levels of functional hemoglobin. Beta thalassemia occurs when a gene that controls the production of beta globin is defective. Beta thalassemia can cause anemia ranging from mild to severe and is more common in people of Mediterranean, African, and Southeast Asian descent. I have the mild form of the disorder commonly referred to as beta thalassemia minor or beta thalassemia trait. Most of the time I don't have any noticeable symptoms, but anytime I have blood work done by a new provider, they initially express concern because my red blood cells are considerably smaller than normal RBCs, and as a result my body makes more of them to compensate for their reduced oxygen carrying ability. I also have skewed liver function tests because my body breaks down more red blood cells than average. This finding often causes providers to question my relationship with alcohol. If you're ever in clinical practice and come across a patient with reduced MCV and high RBCs with slightly skewed LFTs, you'll look really smart when you know the diagnosis. Thank you again for taking the time to view my presentation. I hope you found it informative and interesting. Take care.